Welcome to the first in a series of transplant-focused webinars brought to you by Eurofins Transplant Diagnostics. From donor to recipient, the transplant journey requires the right testing at the right time. That's what Eurofins Transplant Diagnostics delivers. Diagnostics that inform, monitor, and lead to predictive analysis for patients and clinicians seeking personalized insights that can answer life and organ-saving transplant questions. Visit EurofinsTransplant.com for more information. Thank you for attending this webinar.
Welcome to Eurofins session on innovations in transplantation. We appreciate your interest. Although clinical outcomes in solid organ transplantation have improved steadily for decades, median survival rates still range from 5 to 15 years, depending on the organ transplanted. So considerable improvements are possible, and advances in science and technology of diagnostic testing may provide new, powerful tools to help improve these transplant outcomes. Eurofins is committed to leading these improvements through innovative diagnostic testing and research. I'm excited to introduce three cutting-edge researchers who are leading innovative programs in transplantation. I'm pleased to introduce Dr. Prashanth Vallabhajosyula. He will speak on tissue-specific exosomes as biomarkers of transplantation. Dr. Prashanth is a cardiothoracic surgeon who performs the entire spectrum of heart surgeries, including coronary artery bypass grafting, valve surgery, and heart transplantation. He earned his MD at Yale University in 2002, and then joined the faculty at University of Pennsylvania School of Medicine as assistant professor of surgery. Dr. Vallabhajosyula is now assistant professor of surgery and cardiothoracic surgery and the surgical director of the Aortic Institute, which is part of the Yale New Haven Heart and Vascular Center.
Hello, thank you for this opportunity. The title of my talk today is Diagnostic Utility of Tissue-Specific Exosome Profiling in Transplantation. I have no financial conflicts of interest to report. I'll start off the presentation with a case that is very clinically relevant to what we see, especially for me as a heart transplant surgeon. There is a case of one of mine where a 58-year-old female with ischemic cardiomyopathy had a reduced sternotomy heart transplantation procedure. Her case went uneventfully. She had a surveillance endomyocardial biopsy on day 7, which showed no rejection, and she was being planned for discharge over the next couple of days. Unfortunately, on day 8, she developed acute renal failure with acute elevation of liver enzymes and creatinine. An echo was performed urgently, which showed new severe tricuspid valve regurgitation. This was secondary to the endomyocardial biopsy, causing an injury to the tricuspid valve leaflets. On day 9, I took her back to the operating room for reduced sternotomy, tricuspid valve replacement, and epicardial pacemakers. She was discharged on day 28 due to slow recovery from renal and hepatic dysfunction. This is what we face in the clinical setting when we are dealing with patients, and this highlights the importance of why we need biomarker platforms in heart transplantation and in other fields in transplantation as well, compared to biopsies. Complications related to transplant organ injury and immunosuppression remain principal causes of morbidity and mortality in our patients. There's still a critical need for accurate, time-sensitive biomarker assays to monitor transplant organ status and to monitor for the efficacy of treatments. Such a need is most evident in the field of heart and lung transplantation, where still endomyocardial biopsy or lung biopsy remains the mainstay for diagnosing rejection. Let's talk a little bit about extracellular microvesicles and what all that means in the context of transplantation. Extracellular microvesicles encompass a broad range of microparticles that are released into our bodily fluids, including peripheral circulation. These include apoptotic bodies, plasma membrane blebs, microvesicles. Of particular interest to me in my lab are exosomes, which are nanoparticles that are released from the multivesicular body. So these come from the intracellular compartment that contains these secretory nanoparticles that are released, and they're called exosomes. What's exciting about exosomes is they carry specific surface markers on their membrane, and they also carry specific microRNA, mRNA cargos, and protein cargos inside them. Exosomes are detectable in peripheral circulation and other biological fluids. You can see them in culture media all the time. They're well recognized to be a form of intercellular communication now, and they're believed to play an essential role in intercellular trafficking of macromolecules. Exosomes are released by the multivesicular body, as I said earlier, and their quantity and cargoes can reflect condition-specific changes inflicted on their tissue of origin. Here you can see an electron microscopy where you can see these exosomes inside the multivesicular body. Exosomes and their components are quite stable within the peripheral circulation. Studies show that they're more stable than cell-free DNA. Exosome composition may mirror the originating cell and therefore can serve as a surrogate. So some people say exosomes can serve as a liquid biopsy of the tissue of interest. The diagnostic utility of exosomes in transplantation is being increasingly investigated. What's exciting to me in my lab and of interest in transplantation is the fact that exosomes express MHC molecules, both class 1 and class 2 on their surface, which one can imagine could be of great utility for us from a diagnostic standpoint, because it'll allow us to enrich and purify exosomes. We proposed that all transplant tissues and organs release a stable pool of donor tissue-specific exosomes into the blood and other bodily fluids. Because all transplants occur across an iatrogenic mismatch across MHC, this would allow us to actually detect and purify transplant tissue-specific exosomes from total plasma exosome pool. Other donor tissue-specific proteins could also be utilized, such as troponin from cardiac heart exosomes potentially. Also, immune cells themselves would also release a detectable pool of tissue-specific exosomes during acute rejection episodes. Therefore, if we can actually profile the proteomic and RNA signatures of tissue-specific exosomes, i.e. the donor tissue exosomes and the immune cell-specific exosomes, we would have a non-invasive readout of what's happening to the transplanted tissue or organ, i.e. we should be able to differentiate between organ maintenance/quiescence versus rejection/injury. I'll talk a little bit about these models that we developed in our lab and a little bit of results that I'll share about where we're coming from with regards to tissue-specific exosome profiling. We do a standard setup for exosome isolation. We always have been doing size exclusion chromatography as our starting point from a plasma sample. From there, we check absorbance, and then we take the sample from the absorbance peaks that are relevant to us, that suggest to us that we have good separation of particles by their size criteria. Then we do ultrafiltration, ultracentrifugation, and then we put our product on the nanoparticle detector to make sure that the particle size we see fall in the exosome range. All this is basic quality control that is done on all the samples and the results that I'll be discussing today. The nanoparticle detector basically works by looking at Brownian motion, and it can give a readout based on the Brownian motion of particles. It gives us a size distribution. Based on that, we can get some readouts about the particle size distribution and its concentration. What's also nice about the nanoparticle detector is it also has a fluorescence mode that allows us to detect for specific surface markers that are expressed on nanoparticles. So this is the kind of readout that we can get, where you get a particle concentration and size distribution. So based on the nanoparticle detector analysis, let's talk a little bit about the first model that we studied. This is a xeno-islet transplantation model. It's a pretty simple model where we take an athymic nude mouse that accepts all organs and tissues. We give it streptozotocin to induce diabetes, and we give it a critical mass of human islets into its renal capsule. The animal is now normal glycemic, and we follow it out. And using this model, we looked for donor islet-specific exosomes, which are human HLA exosomes that express human HLA. When we looked at this, we clearly noted in our initial studies that in the xeno-islet mouse, we could clearly detect donor-specific exosomes in the mouse blood after transplantation. When we looked at the total plasma exosomes, when we looked at the normal animal versus a rejection arm animal, R for rejection, N for normal, we found that there's really no difference in the total plasma exosome pools during rejection of these in the xeno-islet model. But when we looked at the HLA specific signal representing the human islet signal from the exosomes in the mouse blood, we found a dramatic difference in the HLA specific signal in the plasma exosome pool of the mouse. This was our first window into saying that transplantation rejection would lead to dramatic, potentially significant changes in the plasma exosome pool that contains the specific signal from the donor tissue. This is a dense slide, but bear with me. On the top left, here's a study that we did where we looked at rejection, and we also looked at fasting blood glucose values in animals, and we looked at the donor HLA specific signal. If you could first focus on the red dots here, you will see that we did a xenoislet transplantation and we induced rejection in these animals. The first day we see a stable signal after a transplantation, but immediately, because these animals are rejecting, we see a significant drop in the donor HLA signal in the mouse blood. And that drop continued to come down over time as the animal rejected. What was important is that this drop that we saw occurred even before the animal showed an increase in its fasting blood glucose. We also did serial glucose tolerance tests, and even on those, the donor HLA signal was far more sensitive and accurate compared to glucose tolerance tests. On this model, we also looked at the maintenance arm, which is the black dots, as you can see here, that after you transplant, there's a stable donor HLA signal that is maintained through the follow-up. When we looked at a receiver operating characteristic curve, we noted that only the donor HLA signal from the exosomes was the best marker and most accurate marker to predict rejection in this model. Exosome quantity or size did not matter. Here you can see the histology confirming our findings. As you can see that by day 2, day 3, we start seeing an infiltration of the islet capsule underneath the renal kidney, under the kidney capsule, and we see that there's rejection that's ensuing. And the signal drop in that donor HLA exosome signal occurs before that. We further went on to look at the intraexosomal cargoes. When we looked at the intraexosomal cargoes, here you can see that we validated that when we enriched for donor-specific exosomes using antibody conjugated beads, we can actually see nanoparticles on electron microscopy. We then went ahead and did anti-HLA-A antibody beads and incubated them with the mouse plasma exosomes, and we enriched a bound pool that represented the donor-specific exosomes. And you can see in this model that the insulin signal inside the donor exosomes actually can be tracked and followed. And when you looked at the xenoislet model, there's a faint signal that you could see that occurs in the HLA-A bound fraction. We also cross-validated our findings with another specific marker for islets, especially beta cells, which is FXYD2. And we see that there's a clear FXYD2 signal that we can see in the xenoislet model specific to the donor-specific exosomes. This to us was validation that for the first time, we were actually able to not only detect, but to actually purify and enrich and start looking at the cargoes inside these exosomes that belong to the transplanted tissue. We further went ahead and confirmed that when we actually did a graftectomy of the xenoislet graft, we lost that specific cargo signal here. As you can see on Western blot, the insulin and the FXYD2 signal are gone post-graftectomy, suggesting that this was a specific signal that was coming from the transplanted tissue itself being released into the donor pool. Well, then we said, okay, if this is what's happening with the donor tissue, that with rejection, we actually see a clear drop in the donor-specific exosomes in the blood of the recipient. What's happening to the immune cells? Can we actually pick up the immune cells? Because clearly the rejection process is a dance between the immune system of the recipient and the donor tissue. Of course, other factors are involved too, but those are the two main aspects that we wanted to see. So when we look at the immune cell arm of this, we looked at the T cells. We hypothesized that T cells themselves, because they're revved up and stimulated during rejection, may potentially also release exosomes into the bloodstream. If so, can we track that signal? So we clearly set up methodologies to do anti-CD3 antibody beads and looked at the CD3 exosome signal in the blood. And here you can clearly see, compared to the wild type mice and black 6 mice, which are controls, once you start having the rejection process occur in the xenoislet model, there is a clear CD3 exosome signal that spikes from the day 0 up to day 1, and that signal persists throughout the rejection process. So this is our first window that not only can we start looking at donor tissue-specific exosomes, we could also get a window into the immune system, a little bit about the T cells and what they may be releasing into the bloodstream and what we can track from there. So in summary, just from this set of data, we showed that transplanted islets release specific exosome signal into the circulation. The donor-specific MHC can be utilized to detect, purify, and characterize these exosomes. The cargoes of these donor islet exosomes express tissue-specific markers such as insulin, glucagon, and somatostatin. The transplant islet exosome signal predicted rejection in a time-sensitive manner in this model, and it's definitely more accurate than other current biomarkers such as fasting glucose and glucose tolerance tests. We also noted from these studies that rejection also leads to an increase in the circulating T cell exosome signal that could be tracked and also be utilized as a potential biomarker. We then went on to study the same idea in the clinical setting in human islet transplantation. And we saw that in these four patients, I'm just outlining some of the study, we received samples from the NIH about long-term follow-up on patients who received islet cell transplantation. And here you can see that we clearly saw a donor-specific exosome signal that we could detect the blue lines after transplantation that was persistently detected throughout follow-up. This was pretty exciting to us. Of the four patients, one patient actually had a drop in the donor-specific exosome signal. We found out actually that this was one patient who had autoimmune diabetes recurrence and rejected the graft. And therefore, we validated that concept that at the same time, when GAD65, which is the red line, which is an islet autoantibody, when GAD65 increased, there was a concurrent decrease in the donor exosome signal, suggesting that we may have a window into this immune process just from these blood samples of these tissue-specific exosomes. We went on to further study this in another patient in detail. I'm outlining it here. You can see there was one patient who had islet cell transplantation, accepted the transplant tissue mass, but then had acute rejection and then rejected the islet mass. In this patient, we went on, and if you can see here, these are just validation studies for tissues exosome isolation. But if you focus on part E here, you can see that post-transplant at 10 minutes, we did at 20 minutes post-transplant, we didn't see much of a signal. But by day 2, we started seeing donor-specific exosomes in the bloodstream that we could actually enrich for the islet-specific markers such as insulin, somatostatin, and FXYT2. And we saw that after rejection, that signal is gone. We further validated this by RT-PCR. You can see the same signal that you can actually see insulin, glucagon, and somatostatin mRNA inside these exosomes, suggesting that the concept that we saw in the mouse model could be potentially translated to the human clinical setting. So given these findings in the other transplant models, we sought out to see, can this be translated to other tissue types? And does the size of the transplanted tissue matter? So we studied this in a rat orthotopic left lung transplantation model where we utilized a transgenic rat that carried human CD63 with GFP tag on its surface. So all its exosomes expressed this transgene on its surface. And the recipient was the lowest weighted, a full MHC mismatch left lung transplantation, and looked for this CD3 GFP signal to track the donor lung exosomes. On clinical follow-up, we did daily routine computer tomography and noted that there was clear rejection that we start seeing around day 3 to 4 transition. On histology, we clearly saw infiltration and grade A1 rejection start on day 3 that further progressed to by day 4, and the allograft fully rejected further down by day 7 to 8. If you see this, this is what we see on the nanoparticle detector. You can see that if you focus on the left panel here, you can see the human CD63 GFP transgenic exosomes on the scatter mode display the whole entire exosome profile of all the particles that are seen. But here you can see on the fluorescence mode, we are detecting for human CD63 GFP detection, and you can see a subpopulation express this exosomes. On these exosomes, the controls don't show this, and the lowest exosome showed a very low level compared to the background. In this model too, when we looked at total plasma exosomes, we saw that there was no difference in the total plasma exosome pool or the size distribution. But when we looked at the donor-specific exosome signal, the human CD63 expression on the exosomes in the plasma exosome pool of the recipient, Once again, we saw the same pattern as in the xenotrialar transplant model. We saw that in each of these recipients that rejected the allograft, we saw a significant drop in the donor exosome signal by day 2, day 3 that persisted to be low throughout. And here's a Western blot validation of that finding on nanoparticle detector. Here's a summary of 13 animals that we did where we looked at the donor-specific exosome signal in the lung transplant model. And you can see in all these animals, from the day 1 to day 2 transition, there was a significant drop in the signal. This change also occurred at a time point before there was onset of acute rejection on histology. So similar to the islet transplant model, we saw that there was a drop in the donor-specific exosome signal that happened before there was histologic damage to the tissue. So we saw that in summary, there were very similar results in the rodent models of islet cell transplantation and lung transplantation. We were able to utilize this platform for early time-sensitive detection of acute rejection, and that the donor-specific exosome readout was very similar in both these models. I'll summarize a little bit about the mouse allogeneic heart transplant model that we've done this work on. Here we utilize a simple full MHC mismatch where we transplanted a BAL-C heart into a B6 recipient. The 2 experimental arms were such that one B6 recipient was immunocompetent, where the animal rejected the allograft, and one was an immunoincompetent animal, a skid mouse, that accepted the allograft over follow-up. In each of these animals, we harvested the plasma and analyzed the donor-specific exosome signal utilizing the MHC mismatch. Here you can see we could clearly detect mouse plasma exosomes, and we did all our control studies looking for exosome marker expressions such as TSG101 and CD63 and flotilin in all our studies. If you look at this model, the allograft viability is such that on the rejection model between day 10 to 11, the allograft stops beating. Okay, and in the maintenance arm, we saw viability throughout follow-up up to day 30. When we looked at the maintenance arm, we found that compared to pre-transplant samples, all the 28 recipients that we did the maintenance arm showed a donor heart-specific exosome signal in the blood of the recipient throughout follow-up up to day 30. In the rejection arm, we saw a very similar pattern to the other 2 models, where we saw that on day 1 and day 2, there was a clear signal, but by day 3, that signal dropped significantly and it stayed low throughout the follow-up. When we looked at the total plasma exosomes in this model, the same pattern. There's no difference between rejection or maintenance. But when we looked at the donor heart-specific exosome signal, please focus on the red dots here, the red squares here. You can see that by day 1, there's a clear donor heart exosome signal in the recipient animals. And by day 2, that signal drops. By day 3, it drops further down to almost baseline levels and stays at a low rate. On the maintenance arm, you can see the black dots here. There's a clear signal on day 1, and that's maintained throughout. When we cross-confirmed this with histology on the maintenance arm, we clearly saw no CD3 T-cell infiltration. The allograft was quite viable. But on the rejection arm, you can see by day 5, we saw grade 1R to 2R rejection in the on histology, and that further worsened over time. When we did the same receiver operating characteristic curve, we saw the exact same pattern, whereas the total exosome quantity or exosome size did not predict rejection. The donor heart-specific exosome signal predicted acute rejection with 100% accuracy. We then went on to say, well, what are the cargoes in this model, and what happens between this transition from day 1 to day 2, where this significant drop happens in the signal, even before there's damage to the tissue? So we set up for antibody conjugated bead-based isolation for donor heart-specific exosomes using the MHC mismatch. We basically isolated total mouse plasma exosomes and then incubated them with these antibody beads. The bound fraction represented the donor heart exosomes, and we did an analysis of the cargoes. Here's a Western blot that shows the donor-specific exosome signal that we can track from this model. You can clearly see that we can see troponin inside these exosomes at the protein level and the mRNA level, and track the signal as such, which is really quite reassuring for us that we were achieving good purity of donor heart-specific exosomes. When we looked at the microRNA cargoes inside this, I'm sorry, I have to confess I can't reveal the microRNAs themselves due to some patent concerns from the university side. But I can say that the microRNA cargo analysis showed that there were several pathways that were upregulated in day 2 compared to day 1, including NF-kappa B pathway, T cell signaling, B cell receptor signaling pathways, suggesting that all sorts of allogeneic response elements were being upregulated based on the microRNA cargoes inside the donor heart exosomes. This to us suggests that this interaction between the immune system of the recipient and the donor tissue is far more dynamic, and that there may be physiologic implications here in the immune response itself. This is just affirming that there was a pro-inflammatory network was upregulated based on the microRNA cargoes from these donor heart exosomes. So given that we saw this was altered during rejection, can we actually get a window into the immune system as well? So we start out to do some T cell exosome isolations and analysis in this mouse model. You can see that we use Jurkat cells at first in vitro to establish that we can actually see exosomes from Jurkat cells. And we actually found exosome marker expression, and we found that they were expressing some T cell markers as well. Given that, we also then set up in this model to confirm once again that the donor heart exosome, that the total plasma exosomes are not altered by transplantation in this model, whether there's rejection or even when we do a syngeneic mouse transplantation, B6 into B6, or if we do a full mismatch transplant. We found no difference in total plasma exosomes in this model. But then we looked at the CD3 expressing exosomes in the total plasma exosome pool in this model. When you do a BALB into B6 skid, you obviously don't see any signal for CD3 because there's no T cells. But when we did BALB into B6 model, we saw a clear donor T cell exosome signal, a CD3 exosome signal that persisted throughout the rejection process. But when we looked at the syngeneic model, we saw a signal early on, but then that signal went away and stayed low throughout. You can see that on the Western blot, on the RT-PCR analysis down here, where we can actually see CD3 epsilon mRNA being detected in the donor exosome, in the T cell exosome pool. This is a more detailed analysis of this, showing this. And if you can just focus on the slide on the figure on the right, from the panel on the right, you can see that in acute rejection, which is the red squares, with rejection, we see a surge in the donor T cell, we see a surge in the recipient T cell exosome signal that starts to spike around day 3 to day 4, stays high, and then it comes down and goes back to baseline levels after the rejection process is complete. Given this, we then enriched for recipient T cell specific exosomes in this model using anti-CD3 antibody conjugated B. It's the same concept we used for the donor heart exosome isolation. And using that, when we analyze the bound fraction, and we looked at day 4 and day 7 specifically, only because you can see that's where the real jump happened in the T cell exosome signal in the plasma, we found that there was high expression of many inflammatory markers and T cell markers, including CD3 epsilon by RT-PCR, T cell receptor itself, CXCL10, gamma interferon, IL-2, suggesting that T cell exosomes can clearly be identified during rejection episode, and their cargoes can be purified and studied further. When we looked at further analysis, we found that there was upregulation of many alloreactive pathways when we looked at the microRNA cargoes inside these T cell exosomes. You can see in the day 4 to day 7 cargoes, there is upregulation of B cell activation pathways, T cell differentiation pathways. Many pathways that are involved in programmed cell death were all upregulated, suggesting that the cargoes of T cell exosomes and T cell exosomes themselves may potentially play a role in the allogeneic immune response during acute rejection. So in conclusion, in both thoracic solid organ transplant models, we've noted that transplanted tissues release a stable, detectable pool of donor-specific exosome signal into the recipient circulation. Donor exosomes contain tissue-specific markers such as troponin and that we can see inside the donor heart exosome cargos. Tissue-specific cargo profiling suggests that there are specific changes in the microRNA cargos during the early rejection process. And when we looked at this, we found that in both the donor heart exosomes and in the recipient T cell exosomes, there's upregulation of pro-inflammatory pathways. Further investigation could potentially reveal candidate microRNA biomarkers of acute rejection in this model. Investigation of circulating donor exosomes and T cell exosomes may provide a non-invasive readout of the conditional state of the transplanted organ when done together. In the clinical heart transplantation, I'll just take one more minute to talk about how we've now translated this concept to the clinical setting. We're able to utilize the same methodologies to actually start looking at plasma exosomes in heart transplant patients. We can see that on electron microscopy, we clearly identified exosome-sized particles using our purification methods, and we could track the tissue-specific canonical exosome markers inside this plasma exosome pool, i.e. expressing CD63, TSG101, and flotilin-1. We could also, on the nanoparticle detector, see transplant exosomes from the plasma pool and get a good readout using our methodologies. We then studied 4 heart transplant patients over the perioperative period of the first 15 to 20 days, I believe, where we looked at the donor heart-specific exosomes. The idea of this study was, can we actually purify and isolate exosomes with good quality, number one. Number two, can we detect donor-specific exosomes and clearly track that signal in the transplant model? And here we found that we could track the donor heart-specific exosome signal in this model throughout. You can see when in all these 4 patients, we were able to detect specific isolate plasma exosomes, and it was not, which was expressing flotilin, which is a exosome marker, but didn't contain cytochrome C, which more so represents cytosolic markers and apoptotic body markers. In these 4 patients, we purified donor heart-specific exosomes using the same HLA mismatched concept that we used, that I showed before for the animal models. In all 4 patients, A, B, C, and D, on Western blot up here, you can see that we could clearly track the troponin signal inside the donor heart exosomes. And on when we further analyzed, up here is the RT-PCR, and you can see that we can clearly detect troponin mRNA signal inside the donor heart exosomes throughout follow-up in these 4 patients. And when we did the Western blot analysis in these 4 patients, we could also detect the troponin protein signal inside the donor heart exosomes in all 4 patients in the perioperative period. Of interest to us was this one patient, patient C, who actually developed antibody-mediated rejection in this model, in this one patient, around on a day 8 biopsy. So we hypothesized that if it is true that antibody-mediated rejection that occurs through complement fixation occurs by primarily through antibodies to the donor HLA antigens, then can we detect complement deposition in the circulation itself on the donor heart exosomes, because they're also carrying the donor HLA on their surface. And in this one patient, we found that to be true. We could actually detect C4d deposition on the donor heart exosome pool, suggesting that maybe this methodologies and this concept can be further carried to study other processes such as AMR. Of course, this is extremely preliminary and it's just one patient, but it is somewhat promising that we could potentially pursue this avenue in the future. So in summary, and in terms of future directions, we found that circulating donor exosome signal change heralds early acute rejection in all the rodent models of rejection that we've studied. In these models, the donor exosome signal change precedes transplant graft injury. The development of circulating T cell and B cell exosome platforms may further provide a more complete non-invasive window into the conditional status of the transplanted tissue. We also note that immunosuppression does not prevent detection and characterization of circulating tissue-specific exosomes in the clinical setting. We're now well poised to understand the diagnostic utility of tissue-specific exosome platforms in the clinical setting. Also, mechanistic studies to better understand the role of exosomes in the immune response itself are ongoing in our laboratory. I want to thank members of my lab who were extremely helpful and wonderful in helping us carry through these projects and these ideas, and other members of from other labs, including Dr. Ali Naji, Dr. Wade Rogers, Dr. Shampa Chatterjee, Dr. Okia, and Dr. Rizzi's lab. Thank you.
I'm pleased to introduce Dr. Juston Weems, who will speak on leveraging microfluidics technology to improve gene expression profiling in transplant monitoring. Dr. Weems is a technical director at Eurofins Transplant Genomics Incorporated. He earned his PhD in biochemistry and molecular biology at the University of Oklahoma Health Sciences Center. And prior to joining Eurofins TGI, Dr. Weems was a senior research associate at the Stowers Institute for Medical Research.
My talk today is talking about leveraging the microfluidics technology and how we can improve the gene expression profiling in transplant monitoring. And so we are Transplant Genomics, a division of Eurofins, and the Eurofins Transplant Diagnostics team. And what we have here at Transplant Genomics is a test called TrueGraph. And I'm sure people have heard of TrueGraph, and TrueGraph is a gene expression profile test that is currently in market that helps detect silent rejection, or sub-AR, in kidney transplant patients. So when we talk about gene expression profiling, I think most people's idea goes directly towards biomarkers. And so we want to think about, number one, why do we use biomarkers, and what are biomarkers telling us? So I think most people understand that biomarkers are the cornerstone for precision medicine. And biomarkers are meant to be a prognostic and an indicator of what's happening, hopefully before symptoms begin to develop or become pretty much worse. And so what we're doing with biomarkers is we're taking molecular data of some sort of capacity, could be protein, could be RNA, could be DNA, and we're trying to marry that with clinical information along with tailoring that medical care to achieve the best outcome for a patient. And so oftentimes this is called personalized or precision medicine. And we tend to think of this as being a proactive approach to medicine where we try to preemptively stop symptomatic or an unwanted disease state from occurring, rather than a traditional medicine approach, which is often a reactive state where we've waited until a patient already presents with symptoms, and it becomes difficult to reverse some of those symptoms, and especially in transplant outcomes. So as we think about biomarkers, one of the questions are, how do we develop these biomarkers? And so one way we develop these biomarkers is we start with exploratory studies, often referred to as omics. They could be proteomics, metabolomics, genomics. But we think about identifying protein, DNA, or RNA, and think of it as giving us an indicator of what's to come. And then we take these indicators and we turn them into a clinical assay through development and validation. And then after that, we apply retrospective longitudinal studies, prospective studies, and randomized clinical trials in order to understand whether or not these are commercially and practically useful as far as our patients are concerned and the disease state that we're particularly after. Now, what I want to talk about a little bit today is how we're going to bridge that gap between these exploratory studies and the clinical assay development and validation that comes after that. Because one of the approaches is that we understand is that exploratory studies take a very broad approach into figuring out whether or not we have genes, or whether we have proteins, or whether we have individual markers that are going to tell us whether or not a patient is either likely to or headed on a disease track. And then we develop this into a clinical assay, but oftentimes the exploratory methodology is vastly different than what our clinical assay is actually run on. And so we want to think about how we bridge these gaps, in particular how us at TGI and us at Eurofins are trying to utilize technology and utilize microfluidics in particular to be able to make this jump. So when we think about developing biomarkers here at TGI, one of the things we think about is gene expression analysis. So when we think about gene expression analysis, often this is done by PCR, and using PCR as a tool to sort of give us an indication of this gene expression. So conventional PCR is going to take place in a PCR tube, determining gene expression for all the primary paired genes of interest, all within a single tube. Now, what we understand about this is that it can be a very powerful tool, but one pitfall that we often find in PCR technology as is that low expression genes might be diluted out, and therefore we might be losing or diluting out key performance value when these genes are not highly expressed. And what I mean by that is to give you an example, is if we have 5 molecules that are low expressed out of 1,000, this gives us a representation of about 0.5% chance, and it becomes difficult to identify these molecules or these genes. And so what we've done here is we've partnered with a company called Fluidigm Genomics, and we have leveraged microfluidics to enhance that ratio to give us a better understanding of these low expressing genes, and also a more quantitative approach to PCR and ultimately to gene expression profiles. So who is Fluidigm? Fluidigm is a microfluidics company that is a selective application of pressure to automate assembly of PCR reactions at the nanoscale level. So what Fluidigm does is they use microfluidics to take us from a microliter scale down to a nanoliter scale. And we are using extremely precise loading and mixing of reactions. And one of the things that we find is that we're not using traditional tubes, therefore this eliminates evaporative risk. Also, it's extremely high throughput, and within a single reaction, we can process as few as 144 reactions or as many as 9,200 quantitative PCR reactions. And should they need a ride, we can do digital droplet PCR as well, where we can process as many as 148 reactions at one time. Furthermore, there's a diverse suite of platform methods. As I mentioned earlier, we can use protein, RNA, DNA, and tailor our particular technology to that. And this provides unprecedented throughput and precision that we're looking at. So how do microfluidics work, and what is the underlying tech behind this? So what we see here is representative of what's called an integrated fluidic circuit, or an IFC chip. And these IFC chips show detector inlets on the left side, and we'll have sample inlets on the right side. And basically what's going to happen is that we will load detector inlets on one side, sample on the other side, and the integrated fluidic circuit will actually pull each from each well and put it into a chamber. And it will partition into those reactions that I mentioned earlier, as many as 9,200 reactions. And what it will give us a heat map, and it'll give us threshold cycles as a representative that you see in panel B here, that you can see that there's a number of genes that are activated here, and then we can get a good value of thresholding. So because of this partitioning, we get a better representation of expression molecules within the well. So for example, if we take our original 5 out of 1,000 molecules in a single tube that we just talked about, by partitioning these out, the statistics will show us that one of those 5 molecules will end up in a single chamber. And so we will have 5 chambers that have one of our low expression molecules in there. However, instead of it being 5 out of 1,000 molecules because of the volume and the concentration based upon the single tube, it adjusts that to about 1 out of 10 in a partition, changing the percentage from 0.5% to 10%, making it more likely to be identified and making it more feasible for us to understand what effect that may have. So beyond that, how are these IFCs going to be different for us? So traditionally, we take a 96 well plate, or we take 96 sample tubes, and we add assay, buffer, sample, and master mix all in one. And I think everybody's very familiar with this process. What's very different about Fluidigm is that you actually are mixing the assay and buffer on one side of the IFC, sample and master mix on the other side, and then we're going to allow the liquid automated handler to bring those samples into the center partitions to allow for maximum mixing and efficiency after that. So what are the advantages of IFCs? One, we can miniaturize the reactions. So we go down to 2.5 microliters of master mix. That will give us anywhere between 96 and 192 data points per sample. Traditionally, in a tube for our PCR reaction, we need anywhere from about 10 to about 35 microliters of reaction volume, and so we're significantly reducing that. Second to that, we're automating the assembly. So the liquid automated handler actually mixes the inlets in the partition for us. And what this does is that reduces the error rates that we see because number one, there are fewer bench steps. Number two, the inter-assay reproducibility is enhanced and the intra-assay reproducibility is also enhanced. Finally, and perhaps most important, is that Fluidigm offers open and flexible formats. So depending upon your genes of interest that you're looking for or your targets, they have options to where we can identify 12 samples and we can run them against 192 gene targets. Or conversely, you can run 192 patient samples against only 24 gene targets. They also have advantages where we can run 96 samples against 96 gene targets and 48 samples against 48 gene targets. So we end up with a optimized workflow using substantially less material. And this optimized workflow results in rapid, quick turnaround tests for quantitation that makes this far superior to a majority of methodologies. So just an idea of how this biomark qPCR workflow would be with Fluidigm is that you spend about a half hour on reagent prep, you transfer into the IFCs, and then you load it into the liquid automated handler that's called a Juno that moves the samples into the partitions that I mentioned earlier. This takes about an hour to do so. And then after that, you take that IFC plate and you're going to put it on the actual PCR machine, collect and analyze that PCR data. And so what is traditionally taking oftentimes multiple days to be able to get a patient sample from extraction to output is now taking hours to be able to do that. Now, as far as the practicality goes, all these advantages are important, but the real question is, do we actually see an advantage relative to what we're talking about here, which is transplant diagnostics and gene expression profiling? And so in a couple of publications, people have identified that microarray versus Fluidigm PCR results in substantially enhanced fold changes being able to be detected. So in this particular graph, what you're seeing on the y-axis are fold changes detected by microarray, and those range from basically 0 to about 3.5-fold detection. When we switch over to Fluidigm using the exact same methodology, the Fluidigm fold changes range from 0 to about 13-fold change, indicating a much higher dynamic range for the microfluidics platform and allowing us to understand better the differences in gene expression profiling. Now, importantly for us, what does this mean for Eurofins TGI? So again, as I mentioned before, we have TrueGraph, which is the first of its kind blood gene expression test that is meant to rule out silent rejection or sub-AR for kidney transplant patients. So traditionally, our test has been developed and run on what's called the Affymetrix microarray system. And so you can see an idea of representative genes on the left-hand side, and you can see log fold change of relative gene expression. And the point that we would like to point out here is for microarray is that if we're trying to understand the differences between the genes that are upregulated and the genes that are downregulated, microarray does a pretty good job of telling us that. Microarray also does a really good job of telling us if a gene is on or off. Where microarray fails to really add inherent value is to understand the magnitude of those changes. So you take the data from microarray and you add the quantitative aspect of the microfluidics PCR, and it actually transforms the graph from the left side to the right side. And we really begin to tease out the subtleties of what this means overall between the two phenotypes that we're trying to understand or trying to differentiate between. And so taking this new microfluidics PCR log fold change that you see on the right-hand side, then that question begins to ask what happens as we try to transition this into a differential gene expression and transition this into a classifier. And so when we look at this, you can see on the right-hand side, this is the way that the two phenotypes, representative images of what two phenotypes look like as far as the differentiation on the microarray. So you can see that there are green and there are orange dots that sort of cluster, but they are often overlapped, and it makes it very difficult to distinguish between the two phenotypes, in part because microarray does not understand or cannot register magnitude changes. It only registers more binary changes of yes or no, it changes. And then you can see that transforms when you look on the right-hand side. So we see that we use the microfluidics PCR, and we understand the subtleties of what those changes are actually meaning towards us. And we actually get a natural, very well-developed differential gene expression separation between the two phenotypes, where you're actually seeing the orange separate from the green. Now, understanding that differential gene expression and the way that these things separate is the underlying basis for any classifier or for any output for an algorithm that we try to use to detect two different phenotypes. And so when we see a natural separation or enhanced separation on the raw data, we can understand that the classifier will reflect that in higher performance of all the metrics that are important to us, including NPV, PPV, sensitivity, specificity, and of course, accuracy. So with that, I'd like to thank you for your time and be happy to answer any questions in the Q&A session. Thank you so much.
I'm pleased to introduce Dr. Rohita Sinha. He will speak about the future of bioinformatics and artificial intelligence-assisted decision-making in organ transplant clinical care. Dr. Sinha is the Director of Bioinformatics at Eurofins Viracor. He earned his PhD in bioinformatics at the University of Kansas, and prior to joining Eurofins, Dr. Sinha was a research assistant professor in the Department of Food Science and Technology at the University of Nebraska and co-founder of Metagenomics Analysis, a company focused on bioinformatics.
Hi everyone, this is Rohita Sinha. I'm Director of Bioinformatics at Eurofins Viracor, Lee Summit. So what we're going to discuss today with this audience is about how the future of bioinformatics and AI-assisted decision-making gonna help the organ transplant clinical care. So, I mean, these kind of research, which is going to be, probably there's going to be a little different from the conventional clinical R&D we have been doing so far. And it's interesting to know that how the things are gonna look different when we go into the world of AI and data-driven science. So as you can see, the slide says that the approach which we need to take care when we try to do this kind of projects and science, that we have to increase the width of the knowledge. So it is very important that all the science we do, it's biology aware, but at the same time, this novel science is going to be data driven. So we have to expand the width of the work which we are doing. We have to increase more technology, different kind of technologies which we use to understand the system, deal with the huge amount of data, and try to make sense of what we are doing. At the same time, it is very necessary that we keep the high depth of the knowledge. So high collaboration of the people of diverse capabilities, different sciences coming together. So research depth is necessary, but we have to increase the width of the knowledge. And that is the message we are trying to give here, that it has to be biology aware and data-driven research. So at the same time, generally this question occurred in our mind that what kind of things we can actually tackle using data science and artificial intelligence and high power computing and bioinformatics skills in future. And when we say in future, actually there's a lot of research is still going on right now. So we see that we are using next generation sequencing technology to do the HLA matching, high resolution HLA matching. We are trying to predict the risk of rejection. We are trying to predict how long an allograft can last. We are looking for using NGS to check the infection, viral infection, bacterial infection. We are trying to calculate the degree of immune response of the immunosuppressions which we give to the people. At the same time, there's huge research based on if we can find novel biomarkers for rejection. So there's a lot of things which are already going on. What's going to happen in the future that we're going to mature these things, because There are a lot of challenges also in this field. We know that human population is a heterogeneous population, so a lot of challenges are there. At the same time, that gives the opportunity to us to how to mature this science and how to develop novel things to assist the decision making in future. So a few examples I want to share. As I said, that things are already happening. People in labs are already investing time, money, and resources to do these kind of things. What we are doing at our place is, one example is the assay which we recently came up with called TRACK, which calculates the donor-derived cell-free DNA fraction in the blood samples. And that's a very good indicator of the degree of injury to the allograft. And that requires a massive amount of sequencing of the cell-free DNA. And on top of that, requires algorithms to figure out which population and what fraction of that population is present in the cell-free DNA. So it's a combination of both, like having heavy computing. At the same time, you are coming up with algorithms to figure out or solve this mixture problem and calculate the fraction of donor-derived cell-free DNA in that cell-free DNA mix. So this is one assay which we developed recently. Another example, again coming from our lab, is the TrueGraph assay, which one of our sister company offers, which is based on the gene expression profile of the blood samples. And that is used to calculate the degree of activity of the immune system to show that whether it is leading towards the rejection or not, or everything is okay. So those things are also very necessary for physicians to understand that how much immune system is active and how much damage that can do to the donated organ. So this assay is also based on, as I said, the gene expression profile of the blood samples. And then the algorithm takes care of producing the classifier to compare a normal or healthy system. I mean, relatively healthy when everybody is transplant patient here. So we call it like transplant excellent, called TX, and then compare to the subclinical rejection, which is called sub-AR here. So this is, again, as I said, it's a combination of new omics technology. This is the data, massive amount of data coming here, but at the same time, converting that huge amount of data into a binary classifier of thinking that how we can add and help the clinical decision making using these kind of tools and applications. Another example which I want to share, and at the same time, I would like to encourage everybody to check our poster. This is the abstract number here, 5484, and the title is Application of Linear Discriminant Analysis to differentiate acute rejection and acute dysfunction non-rejection in liver transplant recipients. So as you can see that, as I said, the human population is very heterogeneous. When you get the clinical samples, it is very important that we understand the distribution of the data. So that is the first task generally in our group we try to do before taking up the classification of predictive algorithms or using those algorithms. So as you can see here, what I'm trying to highlight that when we try to see the distribution of the data initially, you see like a typical class overlap problem where all the classes of transplant excellent, acute rejection, and ADNRs, all the samples were pretty much overlapping on top of each other. That's a very, very complex problem to solve because you cannot find any boundary to classify or bin one class of samples and separate them from other class of samples. We tried different methods. You can see this is called principal component analysis. Again, you will see samples overlapping on top of each other. And then again, all the dimension reduction methods were showing the similar profile to us. And that can be really tricky because when you see these kind of distribution, it is very hard to imagine that what classification algorithms we can use here, which can make sense of this data. So most of the papers, most of the work which we quite often see is try to do the dimension reduction using unsupervised method. Just as example, I said, principal component analysis and other. But the moment we saw this kind of challenge here, we shifted our gears and we started focusing on what if we go for supervised way of dimension reduction and see if that can enable the separation of these samples. So here I'm showing that what we got when we started using linear discriminant analysis. What it does actually is like it minimizes the intra-class variation and maximizes the inter-class variation. So it's a kind of supervised way when for each sample, you know the class association. And you can see we got a very clean separation of the data. So what is left then that any new sample, if you can transform that sample into this new space of 2 dimension, there's a very high probability that we can separate and bin them into a unique bin of individual classes. So I'm not taking the thunder away. I will encourage everybody to see and go through our poster, but that gives us really good glimpse that how easily we could develop this classifier once the distribution of the samples were making much more sense, and we could see the clear separation here. So that shows that when we are dealing with the clinical sample, when we are dealing with the human data, how much heterogeneity and how much class overlap is possible, because even the healthy population can be so heterogeneous, and then disease, of course, can be very heterogeneous in nature. So that leads me to discuss that what kind of challenges a research lab which is focusing on bioinformatics and artificial intelligence and try to drive their science using new type of huge amount of data which we get nowadays. So there are several kind of challenges we face. These are the few common challenges which I think is good to share with the people, and then probably may help have a good conversation around that too. So first problem which we quite often see is called class imbalance, where we all know that in a population, the number of diseased people is always less than the healthy people. So if you randomly grab and try to sample that population, majority of the people are going to be healthy. So it is very hard to find those features which can discriminate disease versus healthy when the number of disease sample you have in your hand is really less. So those problems is called like class imbalance problem. Very often we find in the clinical setup. Second is data heterogeneity. As I said, the human population is very heterogeneous. Even the healthy population is going to be very heterogeneous. And at the same time, the disease population can be very heterogeneous in terms of their physiology and the kind of biomarker. That would also depend on the kind of biomarker we're going to use. So these two challenges are there. Other commonly found challenge is like class overlap. That means that when the disease and the healthy samples just look very similar to each other. So all the feature values they have, they just overlap on top of each other. So that's a complex machine learning problem. And as I said, that is very much possible in the clinical research setup too. And the fourth one is class imbalance and overlap. It's like probably the most complex problem we can imagine here, that first of all, you have very less amount of disease data in your hand. At the same time, a major fraction of that data is actually overlapping with the healthy sample type. So that is like a second degree of complexity if we call other types as like the first degree of complexity. So these are the challenges we quite often face when we do this kind of research, but that is the fun, that is the challenge part that we try to tackle and come up with novel ideas to design computational assays. With that, I will come to my last slide, which shows that how bioinformatics and AI operation at Eurofins Viracor is actually gearing up for the future challenges. So we start with having a highly collaborative team. We have bioinformatics scientists in our team. We have machine learning scientists in our team. Mostly we try to avoid any redundancy. So our softwares are designed in a very, very modular way. Our operation is kind of hybrid, something on premise and other things. If it is needed, we can go on cloud and scale up and scale down pretty easily. Similarly, as I said, we have to increase the depth of, maintain the depth of the R&D, and that we try to make sure by having a solid collaboration with biologists and a number of physicians. So this is what our operation is right now, and how we are actually to Viracor is trying to get ourselves prepared and try to contribute to the clinical decision making by the power of bioinformatics and ever-evolving artificial intelligence. Thank you very much. I look forward to your questions, and thanks for listening. Thank you very much. Take care.
Thank you to all of our speakers for interesting and informative sessions.